Everyone. My pleasure to welcome you to the Wednesday afternoon lecture, today to be delivered by Professor Bridget Hogan, entitled The Life of Breath. Very nice use of language there. Stem cells of the adult lung. I was asked to give a brief introduction so that I don't slow down the process of getting Dr. Hogan up here to tell you some very interesting scientific stories, but I will mention that she got her degree, PhD at the University of Cambridge in the UK, did a postdoc at MIT, went back to the UK where she became ultimately head of the Laboratory of Molecular Embryology at Mill Hill. But then she came to the US in 1988 to Vanderbilt, where she's professor in cell biology. However, since 2002, she has been at Duke University Medical Center, where she's now the George Barth Geller Professor and Chair of the Department of Cell Biology. She's also professor in the Department of Pediatrics and the director of the Stem Cell and Regenerative Medicine Program. She's an elected member of the National Academy of Sciences and also of the Institute of Medicine and also of the Royal Society of London. She's a former president of the American Society for Cell Biology. Her laboratory has been engaged in a very productive series of experiments over some years to understand how those resident stem and progenitor cells maintain the complex tissue architecture and multiple functions of the mammalian lung and how they respond to various insults. And she will tell you some very interesting studies that involve mouse genetics, organoid cultures, and genetic manipulation of primary cells, derived all from the human lung. So please join me in welcoming Dr. Bridget Hogan.
Right. Well, thank you very much indeed. It's a tremendous honor to come and give this talk about some of the recent work that's going on in my lab that has been funded by the Heart, Lung and Blood Institute. So as we've been walking around in the beautiful air, looking at the cherry blossom and breathing the warm spring air, I'm really happy that my lungs are in good shape. But I realize that for millions of people throughout the world, this is not the case. And our research is really trying to understand what cells are maintaining the lung and how it helps to repair them after certain kind of injuries and what can go wrong. So the lung is a very complex, beautiful organ system. Like many, it's made up of two major cell types, epithelial tubes that branch finer and finer and finer and end in these multiple millions of little air sacs where the gas exchange takes place. And these are highly vascularized for the gas exchange. And of course, there is a lot of mesodermal cell types around which contribute not only to the vasculature, but to the smooth muscle and lymphatics and fibroblasts. So we've been particularly interested in the stem and progenitor cells of the epithelium. And one of the important things is to remember that there's not just one stem cell type for this system, because the epithelium is very different in the different parts of the lung. Down here in the alveoli, the gas exchange, the very thin-walled sacs are lined by what are called type 2 cells, which make the surfactant, and type 1 cells, where the gas exchange takes place. But up in the airways, the air coming in has to be hydrated, particles have to be removed, and this is affected by a mucociliary epithelium, which is made up of mostly multiciliated cells and secretory cells, which are intermingled, and a few neuroendocrine cells. So there's very different cell types, and they have very different cell biologies and physiologies. So the work from many different labs has led to an understanding of the kind of stem and progenitor cells which are in the human lung and in the mouse lung. Now, there's a big difference in the size between the mouse lung and human lung, but the basic idea is very much the same. So up in the mouse trachea and main bronchi, there is this mucociliary epithelium with the secretory cells and the ciliated cells. And these are maintained by a population of basal cells, which, like basal cells in many other tissues, in the skin, in the prostate, in the mammary gland, are characterized by p63 and keratin 5 expression. And they do not, they're next close to the basal lamina, but don't reach up into the lumen. And these basal cells are present in the human lung, throughout the airways inside the lung, and right down to the very smallest airways. So the very small airways, where a lot of disease happens, as I will show you in a minute, is very similar in organization to the trachea and main stem bronchi of the mouse lung. So we often use this as a model for understanding what's going on in the small airways of the human lung. But down here in the smaller airways, there are very few of these basal cells, and there may be a few cells which have got very low levels of expression of keratin 5 and p63, but they are not obvious from histology. And then down here in the alveoli, as I said, there were the type 2 cells and the type 1 cells. So this is just summarizing a little bit about what we know at present about these stem cell populations. And I will say that it's quite a complicated story, because in steady state in the lung, there's rather little cell turnover. And only one can really see what the potential of these cell types is to do various injury and infection models of the mouse lung. And then we see that if cells are damaged, resident cells can change their behavior and start to proliferate and even change their phenotype. So what has been found is that these basal cells here will self-renew and over time, very slowly give rise to the multiciliated cells and secretory cells by lineage tracing. But recently, Jay Rajagopal in MGH in Harvard showed that actually if you can have an injury model in which you genetically ablate many of those basal cells, some of the differentiated secretory cells can change their phenotype and can go back and behave like basal cells. They will express p63, they self-renew over a long period of time and behave as bona fide stem cells. So this was quite surprising plasticity, which now has been seen in other organ systems as well. And then we showed that these club cells can self-renew over a long period of time and can give rise to multiciliated cells. But a number of labs have shown in certain injury models, injuring the lung with bleomycin, which is a chemotherapy agent that can lead to lung damage in humans when it's given for cancer treatments. Under those conditions, some bona fide club cells expressing markers of differentiated cells can start proliferating and will actually give rise to type 2 cells and to type 1 cells. Again, a sort of fairly unexpected plasticity, which has now been seen in different injury models. And then not long ago, we showed that type 2 cells can self-renew over a long period of time and can give rise to type 1 cells, both in steady state slowly and then after partial pneumonectomy, when there's regrowth and the development of new alveoli in the remaining lung. And under those conditions, when there's a lot of change and remodeling in the lung, even some of these type 1 cells, which are very flat and thin and cover a large surface area, some of them, by lineage tracing, can round up and actually give rise to type 2 cells. Again, quite an unexpected finding. A rather small population were doing it, but it's been repeated in different labs. And then most recently of all, people have been looking at lungs after influenza virus infection. In particular, a PR8 strain of influenza virus, which causes a lot of damage and a lot of inflammation. And under those conditions, Some cells which are still rather poorly characterized in these small airways, which have been called lineage negative epithelial cells or distant airway stem cells, can start proliferating and turn on P63 and keratin 5 and actually migrate out of the airways. With some strains of influenza do this, others don't, but they become migratory, they move out, and they give rise to what are called keratin 5 positive pods of epithelial cells. And it's still a bit controversial, but under certain conditions, Hal Chapman's lab in UCSF has found that after putting these cells into the, transplanting them into a lung and inhibiting Notch signaling, some of them can give rise to type 2 cells. So again, very unexpected, but really generating a lot of data that's quite fascinating and is pointing towards mechanisms for plasticity of epithelial cells that have been rather unexpected. So what we're trying to do is to understand the basic biology of these different populations and to understand the mechanisms driving both their steady state self-renewal and differentiation and how they respond to these different injury models and inflammatory appearance of macrophages and cytokines and so on. So I'm going to focus actually on these basal cells of the mouse trachea area, but then how we've been translating this to studies with primary human basal cells from the human lung. And this is work of two postdoctoral fellows. The work has now actually been published in the work on Tomomi Tadakuro on the role of BMP signaling in the basal cells. She did this work, and it's now been published in development, but she got her own job in Yokohama City University and is now running her lab in Japan. And Xiaogao, this work was published in the Journal of Cell Biology, and she's still continuing this, but will be on the job market soon. So just very briefly, these basal cells, as I said, they're in the pseudostratified mucociliary epithelium. They express P63. Unlike the basal cells of the skin, which is a continuous layer, and actually in the larger human airways, it's almost a continuous layer, these are sort of individual cells here. They express keratin 5. Some of them express keratin 14, but fortunately for us, they express nerve growth factor receptor on the surface, which has made it, we don't quite know why they express this receptor. They don't seem to respond in culture to any of the neurotrophic molecules, but there must clearly be a biological reason. We haven't picked it up yet, but it means it's very easy to isolate these cells by FACS sorting. Now, I said that this is a similar kind of organization in the human lung, and in fact, in the mouse, there are a few submucosal glands in this region, but in the human, these submucosal glands extend again throughout the lung. And I just wanted to include two slides just showing why the relevance, of studying these basal cells to human pathology. Because here is the normal human airway taken from a lung sample, showing here we've got the P63 positive cells and the secretory cells and the expression of the nerve growth factor receptor. So you see there are many more abundance of these basal cells in the human airway. But in this condition of chronic obstructive pulmonary disease, or COPD, which is frequently found in heavy smokers, but in people with lung pathology, there is often squamous metaplasia in which these basal cells will over-proliferate and form multiple layers, and they will lose the ciliated and secretory epithelium, and of course then it can lead to obstruction of the airways. So that's a pretty serious condition. And then also in asthma, I said that in a normal airway, these basal cells will differentiate into multiciliated cells and secretory cells. This is a highly regulated process to keep the balance of these two cell types correct. But in asthma, there's overproduction of the secretory cells producing a lot of mucus. And you can see here, this is from a serious condition of asthma in which there are many of these cells packed with secretory material, goblet cell hyperplasia. And then there's another condition called bronchiolitis obliterans, which is almost the direct opposite. There are far fewer epithelial cells. Sometimes they're, again, not very well differentiated, but there are still definitely P63 positive, keratin 5 positive basal cells in this region. And the paucity of this epithelium can often lead to an overgrowth of the underlying mesenchyme. So one of the things we've learned is that there's a very intimate exchange of information between the epithelium and the stroma, which we have revealed in some of our injury models. So this is why we want to understand these basal cells, and we want to understand the mechanisms by which they give rise to a balanced proportion of ciliated and secretory cells. So what do we know about this mechanism of self-renewal and differentiation? Well, this last schematic is just to summarize what is known at the moment. These are the basal cells, and it's I've kept on saying to you they're P63 positive, keratin 5 positive, and they've got these surface markers. But are they a homogeneous population of cells, or is there heterogeneity among them? Are there some which are quiescent and what people call real stem cells, or are there some which are on the way to differentiating, and even though they express these markers, are somehow got a different genetic program going inside them? Well, some very beautiful paper came out last year from Emma Rawlings' lab in the Gurdon Institute in Cambridge. She was a postdoc in my lab, and she teamed up with Ben Simons, who is a theoretical chemist who's become interested in the population biology of stem cells in a number of different organ systems. And by looking at cell turnover in the normal steady state lung and looking at the lineage behavior of the cells, they came to the conclusion that actually about half of the cells which we were calling basal cells are in fact committed progenitors. They call them committed luminal progenitors, and have got a different gene expression profile and are more likely, have a higher probability of differentiating than the other 50%. So that's quite interesting, and as I will tell you at the end, we think we have identified another components of the genetic differences between the basal cells actually in the human lung, which is predicting those which might start to differentiate towards the ciliated lineage. But then these committed progenitors will start to differentiate, some of them towards the ciliated, multiciliated cells, and work from a number of different labs, not only in the lung, but also in the Xenopus skin and in other multiciliated epithelia, have shown that there's what's called a ciliogenesis program, starting off with multicillin and RFX2 and FOXJ1 are key transcription factors which any of which are mutated will affect the differentiation of these multiciliated cells. FOXJ1 mutants, for example, have very short malformed cilia. They do start the program, but the multicillin is at the top of this hierarchy. And then others believe that Jay Rajagopal's lab thinks that CMIB might be even earlier in the progenitors, giving rise to the multiciliated cells. And then the other program is then going towards these secretory cells, forming the goblet cells or the club cells, as they're called now in the ones which are making these secretoglobins. And we know now from work in my lab and others that high notch signaling promotes the differentiation of these secretory lineages. But in addition to these lineage choices, there's some very, very interesting cell biology which is going on here, which we've tried to see how the change in the behavior of the cells is integrated with their lineage choice. And that is because these basal cells really don't have a clear apical membrane. They have they're attached to the basal lamina through integrins, but they don't have a clear apical domain. And I will show you that we've been interested then in how these cells, as they start to differentiate into either the multiciliated program or the secretory program, now become polarized much more clearly and then form junctions between the cells to form a barrier. And it's this barrier which is crucially important for lung function, because it's keeping out all the microbiome that we're beginning to learn about what's in there. It's keeping out the viruses, it's keeping out the particles. And if this barrier becomes leaky, it's thought that it allows infiltration of things, and it can then lead to inflammation and abnormal lung function. So we've become interested in how you coordinate this morphogenesis of this epithelium. Now, I said right early on that there's very little normal turnover. The lung is not like the gut and the skin, where the stem cells are turning over to replace the epithelium every few days. People have looked at this turnover, and it's very, very slow indeed. There's very little incorporation of EDU into basal cells normally. So we are really challenged to give some kind of stimulus to the lung to get the remodeling going. And this can either be infection with bacteria or with viruses, or one model that we've used quite a lot is to have the mice to breathe in sulfur dioxide, which is sort of mimicking exposure to toxic things in the air. Now, I've been realizing there's a push to include both male and female animals in injury models or studies at NIH. I have to say we do this with male mice because the females don't injure very well, and I've never really quite know why. I think the females all huddle together and hide their noses in each other's fur. They cuddle up, whereas the males probably say, oh, and they're taking big breaths of the SO2, but I haven't really tested that. But we expose them to SO2, it dissolves in the liquid, an acid exposure, then the luminal cells slough off, leaving behind the layer of the basal cells. And most of them seem to survive, and they will start proliferating. You see, here are the P63, here's some non-specific staining of the dead cells, and by 24 hours, most of them have incorporated BRDU and started to proliferate. So there's not a special population that starts proliferating. Most of them seem to proliferate, and then by 2 weeks, this epithelium has been restored, the cilia are beating again, and the mice go back to normal. So this has been a model that we have looked at over the years, and this is the sort of thing that we conclude, is that the surviving basal cells, as I will show you, their first response is to spread. And actually they changed their behavior so that now instead they didn't have any junctions between them, but I will show you that they do now assemble ZO1 positive zonular occludens junctions between them to act as a barrier as quickly as possible. And then we have the undifferentiated progenitors that are produced. Then they start to express lineage markers like FOXJ1, and we get back to having the right proportion of ciliated and secretory cells. There are also changes in the stroma, which include changes in the angiogenesis. There are a leakiness of the blood vessels. There's an influx of neutrophils and macrophages, and changes in the stromal population of fibroblasts that are in here. Now, just very briefly, we did have some funding from the Counteract program to see what would happen if mice were exposed to chlorine gas. And in there, the response is a little bit different because the basal cells are damaged. This was done in collaboration with D. Gunn at Duke, and his student had shown that more of the basal cells die under this condition. And then in about 50% of the mice, the stromal cells overgrow. Their proliferation is not suppressed by the proliferation of and behavior of the basal cells. And in this case, there is stenosis of the airways. And we think that this could be a sort of model for a bronchiolitis obliterans-like pathology in the human small airways if the basal cells can't somehow defective in being able to proliferate. So what we wanted to do was, part of our general program, is to find then factors that will promote the proliferation of basal cells so that you might give this as a therapeutic agent after exposure to toxic chemicals, or just in general to say what is promoting the proliferation and what is controlling their differentiation into ciliated versus secretory cells. So we set up a number of years ago, Jason Rock, a postdoc in my lab, set up this organoid assay. Actually, it was before organoids. It was about the same time that people were doing intestinal organoids, but it was one of the first organoid three-dimensional culture systems. And we set it up as a way of screening for molecules that would promote proliferation and differentiation into ciliated versus secretory cells. So what we did is we took the mouse airway, large airways, dissociated them, and did a pretty, it's a very simple FACS sort based on nerve growth factor receptor expression. And they're about 90% basal cells. We seed them into Matrigel in 3 dimensions in these little inserts, and then put them into a culture medium here. And over the next 2 weeks, single cells, we did a clonal analysis saying single cells will give rise to a little sphere, which we call bronchospheres or tracheospheres. And then I'll just see if I can show this movie. Okay, the single cell, it will form a little cluster, and eventually you form a single central lumen that will pump fluid. So and then if you take a section of one of these, you'll see the basal cells around in contact with the matrigel and the keratin 8 positive luminal cells inside, and then they will differentiate into secretory cells, which are making this protein called plunk. And the multiciliated cells, which the cilia will beat, are functional, and they're marked here by acetylated tubulin. And something was published that Tomomi did in a screen was that we actually isolated basal cells from mice which are transgenic for a reporter GFP driven by the FOXJ1 promoter. So all the ciliated cells would be green, and she could screen then for spheres or for wells which had more green cells in them. And in this way, she identified IL-6, the cytokine IL-6, as a molecule which would promote the differentiation of ciliated versus secretory cells. And actually, this assay was used by Novartis, by Aaron Jaffe, who had been somebody who'd been very interested in polarity of epithelial cells. They used this as a very, very high throughput screen for new drugs or for drugs that would actually inhibit or promote the differentiation of secretory cells as new possible anti-asthma drugs. So we were looking for things which promoted ciliogenesis, and they were looking for things that were inhibiting secretory cell differentiation. But you can see the utility of this kind of primary cell organoid assay. So what Tomomi did was to then use this assay, but rather than look at ciliated versus secretory cell types, was just to look for compounds that would promote colony forming efficiency and cell number. Because what you can do is you can take the spheres in one of these wells and you can dissociate them and then count, in fact, sort them again and count basal versus secretory and count just total cell number. So she did a screen. Of course, there were many more molecules than this. I've just put on here those which were particularly good at promoting colony-forming efficiency or cell number. And top of this list was a molecule called LDN 1931, something, we'll call it LDN. And this was promoted cell number and colony-forming efficiency very effectively. There were other molecules in here. There are sonic hedgehog agonists. There are TGF-beta inhibitors, but they were not as effective as this LDN, which is an inhibitor of BMP signaling through BMP receptors and through SMADs. So as many of you know, of course, the BMP canonical pathway is going through phosphorylation of SMAD1, 5, and 8, which links up with SMAD4 and goes and promotes or represses certain downstream genes. And LDN was an inhibitor of this canonical pathway. Now, there's a little bit of inhibition of the VEGF pathway for this drug. So we were very fortunate to have some help. I'm sorry, I've left out a collaborator from Vanderbilt who had made other molecules which you can now buy commercially called dorsomorphins. And dorsomorphin is very similar to LDN, and it inhibits the BMP pathway much more specifically. So some of the studies I will show you were done with dorsomorphin as well, but this has exactly the same effect. So what we found was that dorsomorphin would actually promote, maybe I've left that slide out. Oh, no, it's here. Okay, so this is the control, the size of the spheres. You can see them here. With dorsomorphin 1, dorsomorphin 2, and LDN, there were more spheres, and they were bigger. But when we sectioned them, Tomomi found that they still had the same proportion of ciliated and secretory cells. So unlike the IL-6, it wasn't affecting the fate decision. It was just affecting proliferation of the cells. And then we found, which is actually not on here, but a little bit later, that if we added BMP, then we got fewer spheres, and they were smaller. So then Tomomi looked to see was this affecting proliferation, and was it affecting differentiation. And so the spheres with added BMP were much smaller. There was much less proliferation. And in this case, all of the cells remained P63 positive. So although there was less proliferation, they stayed as stem cells, and they didn't differentiate. But with added dorsomorphin, there was more proliferation here, more EDU incorporation. The spheres were bigger, but they had luminal cells, and they had the same proportion of ciliated and secretory cells, and they had basal cells around the outside. So it seems that the BMP is inhibiting cell proliferation, and maybe then we're looking into this now, whether it's slowing down the cell cycle, and therefore it's slowing the differentiation of these P63 positive cells into committed progenitors. And then the dorsomorphin is speeding up proliferation. So now what happens in the injury model? This was our screen. So now we said, well, is there any relevance of this to the in vivo repair of the epithelium after damage by SO2? So we went in and collected both whole trachea, luminal cells and basal cells and epithelial cells and stroma, and did a whole lot of studies, which I'm just summarizing, showing you a key finding here, is that this is in the uninjured, and this is the level by PCR of certain components of the BMP signaling pathway. And Tomomi found that early on after the injury, there was downregulation of many components of the signaling pathway, downregulation of, and these are statistically significant, of ligands, downregulation of receptor levels, But what was striking was that there was an upregulation of follistatin. And follistatin is an endogenous protein that's normally made in the animal, which is an inhibitor of the BMP signaling pathway. It also has some activity against the activin signaling pathway, but in this case, we think that it's affecting the BMP signaling pathway. And by gene expression studies, we showed that it was upregulated both in the epithelial cells and in the stroma. And also the BMP ligands, BMP4, which we had some good reporters for, are also normally expressed in both the epithelium and in the stroma. So then we asked, we showed that there was this upregulation of the follistatin, and that we think that this was promoting the proliferation of the basal cells during the wound repair process. So then we asked, well, if we give the LDN or the dorsomorphin to the mice after the injury, can we promote the repair? Would this be a potential way of treating people to promote the proliferation of basal cells after exposure to something toxic, or people who are at risk for bronchiolitis obliterans? So what we did was to, first of all, count the number of cells that were present in the tracheal epithelium at different stages after the exposure to SO2. And this led to quite a surprising finding. I think we'd sort of looked at the repair process and drawn these little diagrams, but we hadn't looked really, really carefully. And what we recognized was that when it started off, there was this cell, either cell number per unit length of basal lamina, or there was this total cell number in the tracheal epithelium. And obviously, after the injury, the total number fell. But then as it repaired, there was an increase over about, so the cells became more dense. They became, transiently, there were about twice as many cells present per unit, and it wasn't just everything had contracted, because the total number also changed. So, and then gradually, it went back to being the steady state level again. So if we looked at this peak time about 5 to 6 days after the exposure, you can see that there's a piling up of the epithelial cells in the trachea. So this is what it normally looks like, and this is what it goes back to after 2 weeks. But there is this transient piling up. So when we gave the LDN, it did indeed speed up the repair process. We got more cells after 4 days, but by 2 weeks, it had fortunately gone back to being the normal steady state level. So that it didn't lead to a continuous hyperproliferation of the epithelium. So this intrigued us, and we wondered how this epithelium goes back to being the normal density. And there had been reports in cell culture and in a few model systems by Jody Rosenblatt and others about a process of active cell extrusion from epithelial sheets. There's both active extrusion of living cells and extrusion of dead cells. We only looked at extrusion of dead cells in the modeling that we did, but this hadn't really been observed in a kind of a wound repair situation like this. So what Tomomi did was to take the tracheal epithelium at this peak time when we think that the extrusion was starting, and to image it over several hours in a confocal microscope. And a trick was to keep the epithelium really flat. And then she added a drug that stains the cells with active caspase 3, stains them green, and then to take little movies. Well, actually, this was done with fixed tissue, when you can, and then this is done with Nocvu, which is a drug which stains the dead cells in a living tissue. and then to image the cells and to see what was happening. And so this is a movie here showing, it's going to repeat itself, but it's just showing, I hope you can see, that this is some cells which are being pushed out of the epithelium. This one is going to go floating off, and then other ones will pop up and get floating off. And then if you look at after fixation and you look in different levels, this is a cell coming out. And if you go down in different levels, you see the cells that were next to it that have now got phalloiding rings, tight rings here, F-acting rings for thinking that they're contracting as they're pushing out the cell. So, I mean, obviously this is, and this is various stills from the movie, and then this is another fixed tissue showing the caspase staining of the cells which are, again, near the surface and would be likely to be being pushed out. So we think that there's a natural shedding which is taking place to restore the normal cell density. So there's quite an interesting question about what controls cell density. How does it know it's got back to the right numbers of cells, and how are the cells being pushed out? And it gives a slightly different view of thinking about the histopathology of some conditions, say in asthma, where there's a lot of shed cells that people have seen as rather pathological. In fact, it could be a sign that there is a good repair going on and that regions of epithelium have recovered and have pushed out excess cells, and isn't necessarily such a bad thing. So this is just to summarize, which is, I said, I unfortunately didn't put up the reference. It just came out in development, this work. But what we think is taking place during this repair process, we think that normally BMP4 made in the stroma by different cell types and in the epithelium is keeping everything relatively quiescent, and that after loss of the luminal cells, then follistatin is upregulated to inhibit the BMP signaling, and the BMP signaling also goes down, enabling the basal cells to proliferate, and then there's this piling up and cell shedding, and everything goes back to a quiescence. So these are the kinds of things that we're interested in what controls, what is it, is there biophysical properties of the basal lamina that are controlling the cell density. So just very briefly, I want to talk about this second part of what we're doing, which is about the morphogenesis of the epithelium. And again, we have been interested in this repair process in making these progenitors and how they get assembled into something with how these basal cells, how the daughters become polarized and form tight junctional complexes between them as a barrier. And I said the basal cells will actually transiently form junctions between them, and then as the epithelium remodels, it's lost again. So I just want to show you this very briefly. ZO1 expression is a component of tight junctions between cells, and normally ZO1 is only expressed in the luminal cells at the apical surface. And these basal cells, which are marked by P63, have no ZO1 expression. But now, 24 hours after the injury, when there's a monolayer of cells, most of which are P63 positive, you can now see the ZO1 between those cells. And Terry Leckler had made a ZO1 fusion protein, and using that, we can see, if we translate this to this looking down on the epithelium, you can see these beautiful zonular occludings proteins between the cells. And they also upregulate many interesting cell junctional proteins like claudin-4, which, as you can see, is highly upregulated. And then as the luminal cells are made, ZO1 expression goes back to the luminal cells and is lost from the basal cells. So there's a lot of interesting cell biology going on here. And we had proposed a number of years ago that maybe the changes in cell behavior and apical basal polarity and junctional complexes was being regulated by a protein called grainy head like 2, or a family of grainy head proteins, which are highly conserved transcription factors that control epithelial cell behavior in a number of organisms, highly conserved, but particularly in Drosophila, some beautiful work has been done about wound repair in Drosophila ectoderm. These act as dimers, so it's possible to make dominant negative versions of the grainy head transcription factor. And as I said, function had been looked at in Drosophila, but in humans, there had been the homozygous mutants in mouse are very early embryonic lethal, but there are some families now that have been described in humans that have got recessive mutations in grainy head-like 2, and it's associated with deafness, with changes in the skin, but also with asthma. And so maybe there's a hint here that abnormalities in the leakiness of the epithelial layer of the airways might lead to some changes. So what we had done, I don't have time to go into this, but we had made a dominant negative version of grainy head-like 2 by replacing the DNA binding domain with a GFP fusion protein. And using that, we had infected primary basal epithelial cells from the human lung. And we had seen changes in the behavior of the basal cells so that they were no longer able to form tight junctions between the cells, and they were no longer able to differentiate into ciliated cells. And in the course of this, we had collaborated with Tim Reddy at Duke to do ChIP-seq and RNA-seq and microarray studies on either the control primary human epithelial cells or those treated with the dominant negative grainy head-like 2. And we'd found many genes that were changed in expression. The ChIP-seq, of course, had shown that there were thousands of genes which had potential grainy head-like binding sites. But the RNA-seq had shown changes in relatively few hundreds of genes had gone up and hundreds of genes had gone down. But looking at these genes and then testing by PCR for those which had been downregulated in response to the dominant negative grainy head, we came up with many potential targets for the grainy head regulation. I said our hypothesis was that the grainy head, which was expressed in the airway epithelium and the basal cells, during the remodeling process would be controlling a whole set of different genes to do with adhesion, polarity, and making junctional complexes. And indeed, many of these genes that we found being regulated by the dominant negative grainy head were encoding membrane-associated proteins, small GTPases, adhesion proteins, things like claudins, and many components that, which have been found to change in these epithelial cells. And this is just an example. Here is the claudin 4 gene, and this is just showing these very robust multiple grainy head binding sites, and RAB25, which had been shown to be a target for grainy head in kidney cells, again had nice robust grainy head binding sites, and their levels were changed after the dominant negative grainy head. But what we were interested also is this transcription factor, ZNF750, which had been implicated in the differentiation of epidermis from basal cells, and that had these binding sites here. So we wanted to test to see whether these were indeed binding sites, and my student Chris Bockley came to me and said, well, you know, you could test all of these in very quick time using CRISPR technology. So I said, well, I don't know about this, but maybe we'll try 10. And indeed, we got a lenti CRISPR vector, put in short guide RNAs for 10 of our potential downstream targets, and a Cas9 expression here. And so we used this lentivirus to knock out, make mutations in these target genes in primary human basal cells from the lung, which are given to us by Scott Randell in Chapel Hill. So we seeded these cells, infected them with a CRISPR, and then had a control short guide RNA. And after selection, we put them into these two test systems. We either put them into the air-liquid interface culture to say, can they make a barrier epithelium? Can they make junctions that will enable us to have electrical resistance between this layer and the underlying medium? And then can we, if we put them into the sphere culture, will they form spheres? Will they pump fluid? Will they differentiate it into ciliated and secretory cells? We can also look here to say, if they form a barrier, can they differentiate into secretory and ciliated cells? So for proof of principle, we put in a grainy head-like 2 short guide RNA, and you can see that it very well makes little indels in the grainy head-like 2 gene. And so part of the problem here is that we couldn't select out individual clones because it's not possible to expand clones in the medium we had. It's now possible, we could now do this, but at the time we took a population of these cells and tested them. And so as proof of principle, the control short guide RNA will form barrier function, and they would differentiate in this air-liquid interface, but those which have been mutant for grainy head-like 2 couldn't form a barrier. And I will show you that they, so they didn't form electrical resistance here. But even though they couldn't form electrical resistance, they didn't differentiate into ciliated cells, but they did differentiate into mucus-producing cells here. So they are able to differentiate, but not down the ciliated lineage, which is what we had found with the dominant negative. And also, they formed spheres, but they formed fewer spheres, and those which were mutant couldn't form a lumen. But if there was a case where there was still some protein being made that possibly wasn't a mutant protein, then they could form a lumen and they could differentiate. So mutant ones didn't form a lumen. So this system was working. So in the last few slides, I'm going to show you here that we did make mutants in these, I think in the end there were about 8 of them that came through. And this, I want you to focus on the ZNF750, which was this transcription factor we were particularly interested in. We were interested in it because it had been shown to be a component of a complex that was regulating differentiation genes in the basal cells of the epidermis, both promoting differentiation genes and inhibiting progenitor genes. But its function in the lung had not been seen before. So just in this last slide, I'm going to show you that knocking out ZNF750 in these cells delayed formation of a barrier, but electrical barrier, but it did, they did finally form one. But it inhibited the formation of ciliated cells here, and again in little patches that presumably were coming from cells which hadn't been mutated. They did differentiate, but mostly they were, FOXJ1 was down-regulated a great deal, but the mucin-producing cells were not changed. So this led us to say whereabouts in the human lung is ZNF750 being expressed. It hadn't actually been looked at before. And so Shah went in and we got sections of normal human lung and stained them for P63 and ZNF750. And ZNF750 is in both the basal cells, some of them, and in the ciliated cells here. But the interesting thing is that not all of the basal cells were expressing ZNF750. And this here is indicating some which are P63 positive, here, these three of them, but they are ZNF750 negative. And then there are others which are obviously P63 positive and ZNF750 positive. So what we think, and this is our last slide here, is back to our general model of trying to understand the pathways which are promoting these differentiation and morphogenesis. We think that we have identified ZNF750 as being a key player for the differentiation of the ciliated lineage here. And we think that grainy head like 2 is coordinating and orchestrating these differentiation and morphogenetic pathways and these are marking all genes which from the ChIP-seq and RNA-seq we know have got binding sites for grainy head, MIB, ZNF750 now, multicillin, RFX2, and other components of the ciliogenesis program. And so we think that ZNF750, again, is marking some population of basal cells on the way to differentiation. So we are now encouraged by this and the fact that we can now got media in which we can grow these basal cells better. We're going to go in and test other grainy head potential targets for seeing where they fit into this program. So I think this is showing you this potential for using CRISPR technology, obviously now on primary basal epithelial cells from the human lung. So this is, I want to say a particular thank you to NIH because they've given me 28 years of support ever since I came to Vanderbilt. And particularly, I want to acknowledge the support of this Lung Repair and Regeneration Consortium that has funded a lot of the junior investigators. In particular, Chris has got a Junior Investigator Awards from them, and they have been tremendous at promoting collaboration between different labs within the United States. And this is my whole lab. You can see where there's lots of babies, lots of food, and lots of fun that we're having. So thank you very much indeed.
Thank you very much. We have time for a couple of questions, and please do use the microphones. We'll start over here.
Yeah, hi, Bridget. It's Matt Hoffman from NIDCR.
Yeah. Hey.
Hi. I was interested if you know what the pro-proliferative signal is after the FTS signal in your basal progenitor cells. Is it directly coming from the epithelium? Is the FTS binding that, or is it an indirect signal, perhaps an FGF or something coming from the mesenchyme?
We honestly, we don't know. That's a very important question about what is the proliferation. We are assuming it's EGF and FGF, but we haven't actually looked at that.
Okay, thanks.
Okay. Well, a comment and a question. First, yes. Comment on asthma. You spoke of the epithelial thickening, ciliated epithelium on the inside of the bronchus. However, it's a little bit more complex than that. We've got smooth muscle thickening, we've got basal membrane thickening, you get fibrosis, and you end up getting a progressive rigidity and narrowing of the airways. So you've really got a progressive disease with a lot of other things going above and beyond what the stem cells might be doing.
Yes.
Okay, now the question. The question relates to another disease, bronchopulmonary dysplasia, which is common in neonates who get oxygen. This seems like the type of thing that would be perfect for investigating that and modes of therapy. Do you know of animal models, and have you thought about moving in this direction?
Well, obviously, I've simplified everything about asthma. It's really complicated, but I'm just trying to say that this organoid screen has been being used for looking for new drugs. For bronchopulmonary dysplasia, actually it's a really important question that nobody has looked at it very well in human neonates. It's all based on what's happening in mice. And actually in mice, as I said, we don't have these basal cells down to the small airways. And nobody knows when basal cells first appear in the human lung. I think I'm correct in saying this. Because and that would be important because they are the stem cell for repairing the small airways and for the growth of the airways after birth. And I don't think people know when they first appear in the human lung. In mouse, they actually appear just at the time of birth. The airways in the developing human lung are actually all ciliated to begin with, and then and they are all P63. And then P60, I'm not even sure now whether ciliated and P63, but after a time, many of the P63 disappear and they're only confined to a basal layer. So those basal P63 keratin 5 cells don't appear until after birth in the mouse. When they appear in the human, it's not known. So there's a lot to be learned. Yep. You just need better models, monkey models and...
Just to comment, there have been some studies on asthma in terms of doing biopsies. These are done in Europe, not here, in neonatal asthma to see when the first changes are ever seen. And I think you could find this in the guidelines of asthma written by the NIH. And I think that the first changes aren't seen until they're 2 years of age or 3 years of age in symptomatic infants. So that might be correlating with when the stem cells starts appearing, because the stem cells are probably making the reparative things and the changes in the epithelium and all that, and that might be a magic number that might correlate with when all this stuff is happening. You might want to look at that literature, just look at biopsies and asthma. There's very few papers doing this because it's unethical. Thank you.
Yeah.
Richard, let me ask one final question. We actually have an interest group at NIH that's looking at single-cell biology now that one can actually do transcriptomes of individual cells.
Yes.
And I couldn't help but think of that, looking at some of the very complex types of cells that exist in the human airway.
Yes.
Wouldn't it be interesting, in addition to be able to look at individual proteins by immunofluorescence, to basically get a repertory of what's happening in individual cells?
Oh, yes, but it's being done. It's being done. It's being funded by NHLBI. There's something called the lung map.
Yeah.
People are isolating single cells from human lungs, doing transcriptome. Again, I talked about heterogeneity in the basal cells. There's also heterogeneity in the type 2 cells, which are a stem cell population. And in looking at diseased lungs with fibrosis, again, heterogeneity at the single cell level. So it's being done. It's being funded by NHLBI. Cincinnati, a lot of places are just pouring out the data. And what's needed is actually being made accessible. What we need is bioinformatics people to make sense of this huge amount of data that's being produced. So Jim Kiley, I think, would love to tell you about this, and Carol Blaisdell has been tremendous in promoting all of this. So it's being done, yes. And we've done single cell.
Well, there is a reception in the library. Please come and continue the conversation if you'd like to. And meanwhile, let's thank again Professor Hogan.
Well, I'm